Now this brings us to part 2 of the Intro to Medical Genetics lecture, where we'll talk about how are genes replicated and passed on, not at the molecular level per se, but in a more broad-reaching approach. So in the previous lecture, we talked about what makes up the cells in the nucleus and the genetic material within cells. Now let's talk about how these are passed on and replicated from generation to generation. So the propagation of genetic material, it really occurs in 2 different forms. And the first is called mitosis, and the second is called meiosis. Now mitosis is specifically for those somatic cells or those non-sex cells, and while meiosis is for those sex or those germ cells, egg and sperm cells, okay? I'll refer to them as gametes. So let's do an overview of mitosis and meiosis, and then we'll get into the weeds. So mitosis essentially results in 2 daughter cells that carry 2 copies of all the chromosomes. So it's an exact copy of the parent cell. Okay, these cells are diploid, meaning they carry 2 copies of all the chromosomes we talked about in part 1, including the X and Y or the X and X for males and females respectively. And this is in comparison to meiosis, which is that gamete specialized sexual reproduction. What that results in is daughter cells, 4 of them, that carry half the genetic information as the original parent cell. And they carry half the genetic information so that when 2 gametes eventually come together, then you'll have the full complement of chromosomes that we generally think of. So what about mitosis? Mitosis, let's start with the cell cycle that preps for mitosis or meiosis, and then we'll get into the specific phases of mitosis. So the cell cycle starts with the G1 phase, where essentially all the bits and pieces, the proteins, everything except for the chromosomes or the genetic material are replicated. And this whole area is called interphase from G1 to G2. An interphase is basically that prep, that get ready time for mitosis, which will occur later. So in G1, again, it's that replication of all those bits and pieces within the DNA, within the cell, except for the DNA. And then it goes on to the S phase, where it actually replicates the chromosomes. Okay, so it duplicates them. And then the G2 phase is the kind of pre-check phase before we start mitosis, and make sure everything's all right, and make sure everything's ready, and to make sure there's no glaring errors, such as trisomies or other errors that could be detected and would lead to a essentially unsuccessful mitosis or meiosis. So after G2, that's when you begin to enter in to the phases of mitosis. Now let's talk about those specific phases of mitosis. So remember, within the cell cycle, we're at G2, and that's putting us into the first individual phase of mitosis, which is called prophase. And in prophase, we have a few things that are happening. The chromosomes are condensing. Okay. The spindle fibers, which are these little spider-like arms, are starting to extend from the centromeres. Additionally, the nuclear envelope, as you can see right there, it begins to break down. And also the centrosomes themselves begin to move to opposite sides. So centrosomes move. Okay? And that prepares us to move on to the next phase of mitosis, which is called pro-metaphase. And in pro-metaphase, what's happening is that the chromosomes are continuing to condense into their finally tightly packed form, so final chromosome pack. Additionally, there's these things called kinetochores that begin to appear at the centromeres, and those facilitate the pulling apart of the actual chromatid to each side of the cell, which we'll get to in the next one. And then the mitotic spindles come out of the centromeres and they actually begin to attach. So the spindles attach, which gets us ready to move to our next phase, which is called, if it was pro-metaphase before, we're now at metaphase. And in metaphase, you can see one of the main things that happens is the chromosomes align in the middle of the cell. And then additionally, now the spindles are officially attached to a given chromatid sister. Okay, and so a clarification here is that individually, right, we talked about this, there's 22 chromosomes, and then there's 2 sex chromosomes. So this is a chromosome itself, a chromosome. But when it's attached in the figures like you see here, this whole thing here is now called a sister chromatid. And so that's where you'll see some of the differing words here. But basically, when it's a pair of chromosomes, it's called a chromatid. And when they're pulled back apart, we'll call them chromosomes, which is about to happen in the next phase, called anaphase. And as you can see in anaphase, the centromeres actually split in two, so they're pulling them away. Sister chromatids now become chromosomes, and they're being pulled apart to the opposite sides. Ends. And then the spindle fibers also assist in beginning to elongate the cell, which you can kind of see here as well. The cell is actually starting to go from less of a circle, which we saw in prophase, to more of an oval shape here. And that pushes us on to telophase. So telophase. And in telophase, it's basically a redoing of some of the processes that were done in the beginning. So as you can see here, the nuclear envelope begins to reappear. So it comes back. The chromosomes themselves actually arrive to the opposite sides of the cell. The mitotic spindles themselves start to go away. So spindles start to go away. And then the actual spindles and centromeres continue to push the cell apart. So it's beginning to kind of exert some sort of force that's starting to split the cell apart. And what happens is we get to cytokinesis. And in cytokinesis, you can obviously see there's a pinching of the cell wall to create two daughter cells. We started with what we could call a parent cell here. We create two daughter cells that have the full complement of genetic material, which is mitosis. So you end up with two that have all the same genetic material as the beginning parent cell. So these are copy 1 and copy 2. And that is mitosis. Now, meiosis is very similar with a few exceptions. And so the reason I say it's very similar is because the phases, which you'll see here, are the same exact words that we just talked about. Prophase, where they begin to condense, the nuclear envelope starts to dissolve, the centromeres start to go to the sides, and their spindles come out, to metaphase, where they're grabbing on to the different chromosomes, to anaphase, where they're pulling them to the ends, to telophase, where it's kind of putting back the nuclear envelope, and the chromosomes are decondensing, to cytokinesis. But the thing with meiosis is there's two steps to meiosis. Meiosis 1 and meiosis 2. And all this means is that it repeats the process once again, because in meiosis, what you're going to end up in is 4 daughter cells, daughter, make sure I get that right, with half the genetic material. Okay? And again, these are gametes that are being produced in meiosis, and they have half the genetic material, because eventually you'll have a sperm plus an egg to get you back to full genetic material. So that's the difference with meiosis. Meiosis is almost 2 mitoses in 1, but it ends up with half the genetic material within each daughter cell. And I highlight this here in the comparison of meiosis to mitosis. So in meiosis, there are 2 divisions, there are 2 rounds of divisions that we talked about that cause the actual daughter cells to have half the genetic material. Additionally, in prophase 1 of meiosis, there's an actual pairing of what's called those homologous chromosomes that we talked about in a previous question, in a process called synapsis. And what this does is it produces what's called a tetrad. So let's go see what that looks like. We have in prophase here, you can see there's the sister chromatid we talked about, right? So there's 2 chromosomes that are connected to each other. But in prophase 1, you actually have 2 sister chromatids together making a tetrad. Right there. Okay? And so that's important for another difference that we'll talk about next. And with those homologous chromatids, essentially what happens in prophase 1 is some crossing over. And what crossing over refers to is some mixing of genetic material. So let's imagine that we have our 2 sister chromatids. Okay, and let's pick another nice color here. Okay, and let's say this is our tetrad. And what's going to happen is that the genetic material is going to mix a little bit in prophase 1 to give you something that might look a little bit like this. This is a loose representation, but it kind of gets the point across. And it could also look a little bit like this. What you can see here is that there's mixing of the chromatids, and what that gives you is genetic variation, right? Because in meiosis, the point is that you're preparing gametes for reproduction. And that's why when you have children, they don't have your exact same DNA copies. Instead, they have a variation of your DNA. And this is one of the essential processes of how it happens. One other thing to talk about is how is the cell cycle actually controlled in the body? How does it know when to start, when to stop, when it should be blazing ahead in the preparation for mitosis or meiosis, etc., etc. And so how this occurs, as you can see here, is through cell cycle checkpoints, or basically, as I like to call them, red light, green light. If you have kids or you have little nephews or nieces, you'll probably have played the game red light, green light. And what that means is that the body has these complement of cell cycle check proteins called cyclins, and they work to make sure that the processes are going when they're supposed to go, and they're stopping when they're supposed to stop. So you'll have times of cell growth where these cyclin proteins will be involved promoting cell growth. You'll have times of DNA synthesis in the S phase where this cyclin protein is phosphorylating or not phosphorylating certain proteins to make them work. And then finally, the preparation for the G2 checkpoint before it goes into mitosis or meiosis. Those are controlled by different cyclins as well. This one's specifically called cyclin B. So the point here is that cell cycle control is tightly regulated, and generally genetic variations or dysfunctions in these critical proteins will lead to non-propagation of those cells. So let's do a knowledge check. A mutation in the gene for a cyclin protein might result in which of the following? A cell with additional genetic material than normal, so more genetic material than normal. Cancer, a cell with less genetic material than normal, or any of the above. So the best answer here is likely any of the above, because I didn't really give you any specific information about the mutation. You could have a mutation that causes some sort of protein that's supposed to stop a phase. So say you had a genetic mutation in cyclin B, and cyclin B stops G2 from going to the M or the mitosis. If you have a mutation in here that prohibits it from working, what you're going to have is an unfettered and non-regulated access, or at least transition from G2 to the mitosis. Okay? And that could lead to cancer, right? That's uncontrolled cell growth, so A and B are very similar. You could have a cell with less genetic material. So for example, at this G1 checkpoint, let's say that cyclin E is involved here. If you have a variant in here that doesn't allow the cyclins to tell the cell cycle that it's okay to go from G1 to S phase, you're going to have a lot less DNA synthesis, and you're likely going to have cell death. So that would be C. So again, the best answer here is any of the above. So what about inheriting DNA and genes? So this little happy fella here, remember we talked about Gregor Mendel. This is a little figure of him jumping around. I should put some pea plants on there. But we're going to talk about 3 modes of inheritance today. We're going to talk about autosomal dominant, autosomal recessive, and X-linked. So let's start with the dominant inheritance. So dominant inheritance really refers to a disease that is caused only by one pair of genes not functioning, or one gene not functioning well. So what this looks like is you could have an affected father. And let's say that the poor functioning G is the big letter D. And the little functioning gene, the little letter D, means it functions perfectly fine. So you have an affected father, he carries one of the poor functioning Ds. But the mother has both little d's, meaning that she doesn't have any poor functioning of this gene. And what this shows you here is what is the percentage chance that they have a kid that carries the disease or carries at least one big letter D. What you can see here is that there's a 50% chance that they will have kids that will be unaffected, so they'll be recessive, they have little d, and little d, little d. And then there's about a 50% chance that they'll have kids that carry one of the dysfunction alleles, and they'll have the disease. So what does this actually look like in something we call a Punnett square? So let's do that right now. So Punnett square is a way that we can actually tease out genetic inheritance. So let's do the big D and the little d, and the big D and the little d. And this is from dad, and this is from mom. So what are chances here? Just like the figure over there, but a little bit different because we have 2 affected parents in this figure. You can have big D for this kid. You could have a big D, little d for that child. For this child, it'd be big D, little d, and then a little d, little d. And what I'm doing, I'm just crossing the squares. Big D, little d comes here, little d. And so what are these called in the dominant inheritance model? They're called homozygous dominant, heterozygous dominant, the same with this one, and then homozygous recessive. So which ones have the disease? Anybody with a big D. So you have disease here, you have disease here, you have disease here, but luckily you don't have disease here. So with a mother and father that are both heterozygous and are disease carriers, they have only a 25% chance of having an unaffected child. While they have a 75% chance of having an affected child, they have a 50% chance of having big D heterozygous child, big D little d, 25% chance of having a homozygous dominant, and a 25% chance of having that homozygous recessive. So again, if both parents have a disease and a dominant inheritance model, the likelihood that they'll have an unaffected child is quite low. What are some examples of dominantly inherited diseases? Two of the main ones we think about are Huntington's disease, which essentially is a defective protein within the brain, and what you see with that is the common chorea, or the kind of uncontrolled movement of limbs. I think I have an actual video of one here. As you can see, her purposeful movement is very difficult. It'd be very hard to write. All those types of things comes with Huntington's disease. Another example is autosomal dominant polycystic kidney disease. What basically that is you have a normal kidney here, right? But in polycystic kidney disease, you have a bunch of holes in your kidneys. And what that causes is it makes it look like a sponge. And when you have a spongy kidney, it can't do very well. So these are mutations in PKD1 and PKD2, and they're inherited in a dominant form, meaning that if you have a parent that's a carrier, again, like we saw previously, if you just have one parent that's a carrier, you will have a 50% chance of a child having that disease. And again, this is just a figure here showing you that you have a bunch of cysts that causes holes in your kidneys, and it makes them function very poorly. So let's do a quick knowledge check. Based on the dominant inheritance Punnett square, how would you describe the following genotype? Big J, little j. So big J or little j allele status predicts the drug metabolizing capacity of a common enzyme. The big J has reduced metabolizing function because it's a dominant scenario that we're talking about. So answer A is homozygous dominant with reduced function. Answer B is heterozygous with normal function. Answer C is homozygous recessive with normal function, and answer D is heterozygous with reduced function. So let's just do the Punnett square real quick here. So we have big J, little j. So let's pretend that we have two heterozygous parents. Again, big J, big J, big J, little j, big J, little j, little j, little j. So the one we're interested in here, or at least for this answer, is here. And so we know what? One, they're heterozygous. These two are heterozygous. This one's homozygous, and this one's homozygous as well. And because they're carriers of the J allele that has reduced metabolizing function in a dominant model, we know that it's heterozygous with a reduced function. Heterozygous with normal function is not correct because this again is a dominant model as opposed to a recessive inheritance model, which we'll talk about right now. So in a recessive inheritance model, what it happens is that you have to essentially have two alleles of the dysfunctioning gene in order to have it because it's recessively inherited. You have to have two alleles here. And why this is because you only need one functioning copy to not have the disease. So in this depiction here, we have an unaffected mother and father who are both heterozygous. And they have basically a 25% chance of having a child that's unaffected and not a carrier, so they don't have any little r. They have a 50% chance of having a child that has at least one little r, so they're considered carriers, but they don't have disease. And then they have a 25% chance of having a child with both little r's, being a homozygous carrier and having the disease. So let's look at what the Punnett square would look like in this one. Let's do the same thing we did before. We'll do our squares. Alright, so let's do big R, big R, little r. So this is exactly what we're doing over here. So big R, right? Big R, little r. Little r, big R, little r. And then little r, little r. And so what these are called in a recessive inheritance model are a little less complicated than the dominant one. We just call these ones homozygous because they're not a carrier. They're just homozygous. This one's called homozygous affected. This one's called heterozygous, carrier. And that one is 2. So, and all just like over here, 25% chance, 50% chance, and a 25% chance of being a homozygous, non-carrier, heterozygous carrier, or homozygous affected. Okay? And you could do this with any Punnett square. We could change them to a little r parent here. We do a big R parent here. So Punnett squares are pretty easy to do. They're pretty versatile, and you'll work a little bit on your activities using Punnett squares. What are some examples of some recessively inherited disease? The two main ones we think about, especially in the medical community, are cystic fibrosis, which essentially is a mutation in the CFTR gene, which causes your protein, CFTR, to be dysfunctional. And so what happens is you have mucus buildup in the lungs, okay? And so these folks generally get a lot of infections, they have a lot of diseases, and as you can see, they have many more organs than just lungs affected by it. Another common recessively inherited disease is sickle cell disease. And essentially what this is some sort of mutation that causes your red blood cells to not be round and happy like this, but to be sickled. And by sickled, it just means it's referring to a sickle shape. And so what happens is when you don't have properly shaped red blood cells, they'll cause traffic jams, and they'll cause a lot of problems, a lot of pain in these patients, and the potential for other blood-based issues. Finally, we have X-linked carrying, or X-linked inherited propagation, inherited mutations, or X-linked propagation of genetic material, I should say. And finally, we have X-linked propagation of material, genetic material. And basically, this just means that the gene is carried on the X chromosome. So, in this example, we have an unaffected father. Okay, they don't carry the mutation, which is signified by this 1 right here on the X chromosome. So you have an unaffected father, and you have a carrier mother, but she doesn't have the disease. And so what are the chances of them having children that have this X-linked disease? Well, they could have a girl that does not have the disease, or could be a carrier of the disease, and then they could have a boy that either doesn't carry the gene at all, so he's not affected, or he has the actual mutation on his X chromosome, and so he has the disease. And so one of the special things about X-linked diseases is that in females, it's very rare to see them in females. And why is that? Because of what we talked about previously, Bar bodies. When they have 2 times the dose of the X chromosomes, they can inactivate the bad one. So in this example, what would happen is they would become a Bar body, and they would keep the good chromosome, so they'd be a carrier, but they wouldn't be affected by the disease. That's in contrast again to males, right? Who only get a single X chromosome. So if they have the actual mutation on their X chromosome, they will have the disease because they do not have the ability to turn this into a Barr body. So they can't do this. They can't go to a Barr body. Okay? So they only need one X chromosome inheritance to have the disease, and obviously that's because they have one chromosome, one X chromosome. One of some examples of X-linked chromosomes, you have Duchenne's muscular dystrophy, which again is a muscle-based disease. And then you have red-green color blindness. So if you can't see this, you might have that X-linked disease, but the number that's in here is indeed 16, which is right in here. So those are two somewhat common, although obviously muscular dystrophy isn't that common. Red-green color blindness is very common types of X-linked diseases.